Hi students, today we will discuss for the transcription in prokaryotes and eukaryotes, and if you recall our first video, the transcription, it is a process in which the DNA it is copied and RNA is formed. So the copying of the genetic information it takes place in the transcription. Here the enzyme which is responsible, that is DNA dependent RNA polymerase, always polymerizes in 5-3' direction. Here the process it requires a unit which is called as transcription unit, and it consists of the promoter, structural gene and terminator. You can make here the strand that is 3', 5', and you can make here the strand 5', 3'. So this is what? This is the template strand. This is the coding strand. And promoter, it is always present at the 5' end of the coding strand, and at the 3' end the terminator is present. You can label it as promoter and terminator. Now the enzyme in the which is called as RNA polymerase, it transcribes all the 3 types of RNAs in the prokaryotes. Only single enzyme it is present. And the 3 types of RNAs, they are mRNA, tRNA and rRNA. mRNA, it stands for messenger RNA. It provides the template. tRNA stands for transfer RNA. It brings amino acids and read the genetic code, and here rRNA, it is ribosomal RNA, and it is having the structural and catalytic role. So only one type of RNA polymerase it is present, and it transcribes all the 3 types of RNAs having different functions. Now the process is completed in 3 steps. We are talking about prokaryotes right now: initiation, elongation and termination. In 3 steps the process is completed. The RNA polymerase, it binds with the promoter region, and when the RNA polymerase it binds with the promoter region, the unbinding of the DNA molecule it takes place. After that, the polymerization it started. You can see the stretch of RNA, because here the nucleotides they started adding, and the strand it is added with the RNA polymerase enzyme during the course of elongation. And when the enzyme, RNA polymerase enzyme, along with the short stretch of RNA, nascent RNA, it reaches the termination sequence, then the RNA polymerase it detaches so as to the stretch of RNA which is formed in the elongation phase. So in the elongation phase, the polymerization it occurs, where the nucleotides they join together and the RNA strand it is formed. So that is how the transcription it occurs in the prokaryotes. Now one question arises here, that how the single RNA polymerase, it is able to perform all the three functions, like all the three steps, they are being completed through the single RNA polymerase. And the answer is that actually RNA polymerase, it is efficient for the elongation. It is the binding of the sigma factor and the rho factor, which is responsible for the process of initiation and termination. Actually, after the binding of these factors, the specificity of the process, it alters, right? So this is important here. RNA polymerase, it is only capable of catalyzing the elongation. It, it actually associates with the initiation factor, that is sigma, and termination factor, that is rho, to initiate and terminate the transcription respectively, and association with these factors alter the specificity of the RNA polymerase to initiate or terminate. So that is needed to be considered here. In bacteria, one more point which needs consideration, that here the coupled reaction it is. Coupled reaction, what I mean to say, here the mRNA, like that of eukaryotes, don't need any processing. So what happened? The transcription and translation takes place in the same compartment. The reason behind, there is no separation of cytosol and nucleus, it is in bacteria. We know that bacteria, the nucleoid region it is present and the nuclear membrane it is absent. So as there is no separation of cytosol and nucleus, here many times the translation it begins much before the mRNA it is transcribed. So the transcription and translation, they are, or they can be coupled in bacteria. So this is all about the transcription in prokaryotes, and let us come for the transcription in eukaryotes. In the eukaryotes, as we know that the split gene arrangement it is present. What the split gene arrangement it is, that the expressing sequences which are called as exons, they are intervened by the non-expressing sequences which are called as introns. So actually in the eukaryotes, the introns they are needed to be removed, and also capping and tailing occurs, and collectively it is called as the maturation. You know, the mRNA, it is processed from the primary RNA transcripts, and the process it is called as maturation. Now, let us start with the enzyme that is called as RNA polymerase. Unlike that of bacteria, in the eukaryotes, three types of RNA polymerase they are present. RNA polymerase 1, they transcribes rRNA, ribosomal RNA, and they can be of like 28S, 18S, and 5.8S. RNA polymerase third, It transcribes tRNA, 5S rRNA, and snRNAs. snRNAs, they are thought to be the small nuclear RNAs. So now let us come for the RNA polymerase second, because we are done with the one and third. We have seen the function of RNA polymerase 1 and third. RNA polymerase second, it transcribes the precursor of mRNA, and that is called as HNRNA. Its name is heterogeneous nuclear RNA. Where it is present? In the nucleus it is present. That is why, it is said that mRNA, it is processed here because the precursor is present. hnRNA, it is there. The precursor is present and it needs processing. So the mRNA, it is processed from the primary transcripts. This is what the primary transcript, and this process is called as maturation. Now, what happened here? First, you can say for splicing. Let me check for another color. Splicing. In splicing, what happened? The introns, they are removed, because we know that in the eukaryotes, the monocistronic condition is there, and in the monocistronic condition, the split gene arrangement, exons and introns, they are present. So introns, the non-expressing sequences, they needed to be removed, and exons, they are joined in a defined manner. These are the expressing sequences which appeared in the mRNA. So exons are joined in a definite manner, and the enzyme which is responsible, that is ligase, right? Now, additional feature it is there, that is called as capping and tailing. What the capping and tailing is? In capping, an unusual nucleotide, it is added to the 5' end of hnRNA. What that nucleotide is? Methyl guanosine triphosphate, which is also called as 7mg, right? So initially at the 5' end, a cap consisting of 7-methyl guanosine or 7mg, it is added, and the cap, it is a chemically modified molecule of guanosine triphosphate. So that is why we have discussed this, like at the 5' end of hnRNA, methyl guanosine triphosphate, it is added, and the process is called as capping. In tailing, in this process, adenylate residues which can be write in number. They are added at 3' and in a template independent manner, and after the splicing, capping and tailing, now the processing it completes. The hRNA it gets processed and mRNA is formed from hnRNA, and now the processed mRNA it is being transported out of the nucleus, right? So this is the whole process. We have to remember here for the splicing, the removal of the introns. We have to remember here for the capping. Capping is what? Where at the 5' end of hnRNA, the methyl guanosine triphosphate it is added, and we have to remember for tailing. In tailing, at 3' end, adenylate residues they are added. Right? So now the hnRNA it is processed and come to the cytoplasm from the nucleus. And you can see here in the diagram the capping at 5' end and the tailing at 3' end, where the process it is also called as polyadenylation. The adenylate residues they are added at the 3' end. And here the processed mRNA which is being transported from the nucleus to the cytoplasm. So this is what the difference between the prokaryotes and the eukaryotes transcription. Again, the difference it is on the basis of the type of RNA polymerases. In prokaryotes, only one type of RNA polymerase is present, right? And in the eukaryotes, three types. So you will be able to see here clear-cut division of labor. Another feature which is the differentiating point of the transcription of prokaryotes and eukaryotes. In eukaryotes, the processing is needed. The primary transcripts, they are needed to be processed. And in this phase, the hnRNA, it is converted, it is processed into mRNA. And the three processes are there: splicing, capping, and tailing. So now we are done with the transcription, and we'll meet in the next upcoming videos of ours. Till then, have a very good bye.